All right, welcome back to the final part of chapter 8. So in the past parts, we spoke a lot about, pigments and how they're used to actually attract energy, well, sunlight energy and transform it, actually, give it to the electrons in the reaction centers in the photosystems and how that is actually fueled to do this, light dependent reactions and where we generate NADPH and ATP. Of course, remember, and only in actually in non-cyclic phosphorylation do we generate NADPH and ATP. Cyclic, we only produce ATP. So this is very important here to produce ATP and NADPH because then this fuels the production of glucose. Because of course, creating glucose is very endergonic, so we need energy for this. But also, we are creating new compounds, so we need electrons actually. So NADPH is going to be that source of electrons that's actually going to provide us. So now we're going to move into the light independent reactions, which is this carbon fixation, which we also call the Calvin cycle. So the Calvin cycle comes from actually the inventor. So I think his name was Melvin, yeah, Melvin Calvin. He was the one who discovered this, and where he basically said that plants are able to build carbohydrates, and they're able to do this from the first reaction, which is the light dependent, and where we actually get energy and the reduction potential and the pH. And these are basically giving the source of um, ATP, so it drives the endergonic reaction. And we also get a source of protons and actually energetic electrons from this NADPH. So these two important products from the light dependent, basically photosynthesis is helping to fuel the light independent reactions from the Calvin cycle. So again, Calvin cycle was named after Melvin Calvin. We call this also C3 photosynthesis. So if you ever hear C3 photosynthesis, it's the same thing as Calvin cycle. And they call that because the first intermediate molecule in this whole cycle is actually a three carbons. That's why it's called C3 photosynthesis, because the first intermediate is three carbons. So the Calvin cycle, we'll go much more in details about it. But the most key and most important part of the Calvin cycle is when you get the carbon dioxide and you stick it into this five carbon sugar, which we call ribulose 1,5 biphosphate, RUBP. So when we do that, we attach it, and this gives you a six carbon, actually six carbon, which is then immediately broken into three phosphoglycerates. So you get two, three phosphoglycerates. And all of this is done actually by a very important multi-enzyme complex that we call Rubisco or ribulose biphosphate carboxylase slash oxygenase. We call this Rubisco as well. So Rubisco is able to actually take in the CO2, put it in this RUBP, which gives you this six carbon, and then this six carbon is then broken down into two phosphoglycerates. So this is the three carbon, this PGA. And this is where the whole Calvin cycle gets its name from the C3 photosynthesis. So from this three phosphoglycerate, which is three carbons, this is why we call the Calvin cycle the C3 photosynthesis. So Calvin and Krebs cycle, they might look very similar to each other, but you need to remember that one is actually doing a lot of oxidation. It's really oxidizing the stuff. While here, actually, we're reducing. We're trying to actually do this carbon fixation and creating new compounds. While the Krebs cycle is breaking it down and we're trying to break down into oxidize. So always keep in mind they're two different reactions, even though they're quite similar, and but a bit different. So the phases of Calvin cycle, which is the first one that we already spoke about, which is the carbon fixation, and where we get this this five carbon, this RUBB, and we actually get the CO2 and incorporate it there using Rubisco. So then this gives you a six carbon, which is then immediately, literally broken down into this PGA, so which is this three phosphoglycerate. So you get two of them, and then the second part is reducing this PGA into glyceraldehyde 3-phosphate, G3P. So I'm quite sure you guys have definitely heard of G3P. G3P, glycerol 3-phosphate, is actually one of the first products we get when we break down glucose in glycolysis. So this glycerol 3-phosphate that we produce can actually be used then later to combine with another glycerol 3-phosphate to create glucose. So we're creating the intermediates for glucose here, which is glycerol 3-phosphate. So then after that's done, we regenerate this RUBP because this is a cycle. So again, remember, a cycle is the products then become the reagents again. So to do this, we need to regenerate RUBP. So here we produce a lot of actually G3Ps. Some will go for glucose and some will go to regenerate this RUBP. So actually, if we use three carbon dioxides, we get one new G3P. So in order to get one glucose, we need six turns. Because think about this. G3P is three carbons. So glucose is six carbons. So if every three carbon dioxides gives you one 3GP, three like new ones, that means that we need to actually do six turns. Because glucose is six, so we need two G3Ps. So we're gonna go into much more details about this. So I'm gonna explain to you what happens with one molecule of carbon. So when one molecule of carbon comes in, it's gonna basically incorporate into this RUBP, which is then this Rubisco, and then so basically sorry this RUBP, and with the help of Rubisco, the enzyme, it's gonna put the carbon dioxide in this RUBP, which is gonna create a six carbon, which is then directly broken into three phosphoglycerates. So you're gonna get basically these three phosphoglycerates, so two of them, and then these guys will be turned into 1.3, 1, comma 3 biphosphate glycerates, so C3, by adding two phosphates, which are then broken down into this glyceraldehyde 3-phosphate. So this is the G3P that we're talking about. So we're actually creating this, and then basically some will go back into the creation of Rubisco again, and some will actually go into the production of glucose. So to get one molecule of glucose, you need six carbon dioxide. So this is how it starts. Six carbon molecules of carbon dioxide bind to six molecules of RUBP, to Rubisco. This gives you basically then this is broken down and gives you 12 molecules of 3-phosphoglycerates. Because this makes sense because now imagine you have six molecules of a six carbon. So that means you have 36 carbons. So that's why we need 12 of these 3-phosphoglycerates. So these are 3 times 12 is 36. Then these guys are then phosphorylated. So we add another phosphate, gives you 1,3-phosphoglycerate. And these guys then are, what's it called? Transported into glycerol 3-phosphate. So by the way, from 3-phosphoglycerate to 1,3-phosphoglycerate is phosphorylated. And this is where the ATP comes into. Because now we need to actually use ATP to give that phosphate. And we use 12 ATPs because we have 12 molecules. So 12 ATPs are used here. And then this gives you glycerol 3-phosphate. Of course, this is also with the help of NADPH. So this is where the products of the light-dependent reactions are used. So ATP is used here to actually help us create this 1,3-biphosphoglycerate. NADPH is the help to give electrons to give us this glyceraldehyde 3-phosphate. Then we need two of these molecules are brand new. These are then given out. The rest, so 10 molecules, are required to go give us back the six molecules of this ribulose biphosphate to regenerate the six molecules here again. So we created two extra, actually, if you think about it, two extra glycerol dehydrated phosphate because of the six carbons. Just think about this: six carbons gives you so basically gives you two glycerol, two phosphate, two new ones. Well, these guys, the ribulose, we need to reuse it, so we don't use them too much, so they go back. So you get two molecules of this glyceraldehyde 3-phosphate, three Cs. And then these are then used together to give you glucose and other sugars. So this is the Calvin cycle. This also, by the way, all happens in the stroma. So again, the output of Calvin cycle. So we know that we get G3P, majority of it, but we can actually use this G3P to give us glucose, which can be used for, of course, sucrose. It can help us give us sucrose, but it can also help us do starch. So sucrose, remember, is the disaccharide that is both fructose and glucose that helps these sugars be transported. But also we can give this big long polymers or macromolecules like starch, which helps us a lot, in energy storing. So if we think about the energy cycles right now, especially in plants, both of them they have a chloroplast and mitochondria. So both of them they can do respiration and also photosynthesis. And they're very interconnected with each other. Because imagine this, all so when we actually do the first reaction of photosynthesis, we create oxygen, right? We break oxygen because we need electrons. So water becomes an electron donor. So we get water through the electron transfer system because remember, in electron transfer system, we actually give electrons to oxygen that then gives us water. Well, this water is then used up here by the chloroplast to break it, and that gives us oxygen. This oxygen can also be used by the electron transfer system as your final electron acceptor. So this is how these things are very connected with each other. Remember during the citric acid cycle, we also get a lot of the decarboxylation happening there. Well, we release carbon dioxide through this decarboxylation, which can then be, of course, used here to fuel the Calvin cycle. More importantly, is that in the Calvin cycle, we're producing glucose at the end of the day, which can then be, of course, broken down into into pyruvate through glycolysis, which can then enter the citric acid cycle. So you see actually a lot how the chloroplast and the mitochondria are really kind of completing the energy cycle, how they're really intertwined together and how their products can help each other out. It's super cool and how these two systems communicate with each other through these kind of products they create. So one thing about Rubisco, and I don't know if you guys really noticed that, is that if we go back to Rubisco, and the name of it is ribulose biphosphate carboxylase slash oxygenase. So carboxylase or oxygenase, very two different roles. One of them is carboxylating, meaning adding carbon dioxide, and the other one is oxygenating, so oxidizing. Two very different processes and completely different. So that's the issue with Rubisco. Rubisco is a very brilliant enzyme, but it's not optimal, meaning that depending on well basically the workable solutions or the whatever actually the current climate is, the Rubisco will work in two different ways. So if the plant is having a lot of carbon dioxide, it's going to do this carboxylation, so the carbon cycle. But in cases where there's not a lot of carbon dioxide and there's a lot of oxygen, it's going to favor something called photorespiration. So Rubisco has an active site, and whether it actually gets, there's carbon dioxide. If there's carbon dioxide, it's gonna bind to Rubisco, and it's gonna do the first actually here incorporation of carbon dioxide into this RUBP. But if there's a lot of oxygen, it's gonna bind to Rubisco, and it's gonna do something completely else. It's gonna oxidize Rubisco, so it's gonna give oxygen to it, and this is very bad because when it actually oxidizes it, this RUBP, it actually kind of it how can I put it in this way? It gets rid of a lot of very useful carbon dioxide. So this is not the greatest thing because photorespiration really reduces the yield of photosynthesis. So when would do you see conditions where you have more carbon dioxide than oxygen or more oxygen than carbon dioxide? Hot temperatures. Remember, guys? So we said when there's a lot of hot temperature, when there's basically the temperature is exceeding, let's say, 28 degrees, this is very, dangerous because it really makes the plants lose a lot of water. So what is going to happen is that they're going to close the stomas. So once they close the stomas to prevent this loss of water, oxygen is going to build up a lot inside of the plants, and carbon dioxide is not going to be able to come in. So then suddenly inside of the plants, you're going to be a lot of oxygen. And this is going to really push the photorespiration. So the oxidation of RUBP, not the carboxylation of it, which then reduces photosynthesis. So of course, this is quite bad because we need for plants need photosynthesis. They need to constantly keep doing this. So it needs to actually come up with ways to combat this. So this is again the photorespiration. So you have Rubisco here. This is the big enzyme. So depending if you get oxygen, if oxygen binds to it, it's gonna start actually gonna get RUBP and oxidizes it, and then it's gonna really use up a lot of the carbon. But Rubisco, if it gets carbon dioxide, then it's gonna go to the carbon cycle, which is gonna help us create, this G3P that's gonna help us create glucose. So there has to be a certain way to regulate this, or to keep this carbon dioxide always up when especially when the stomas are closed. So certain plants cannot do this. Some certain plants can only basically do photosynthesis, so the C3 photosynthesis, the carbon cycle, and there's no way they can combat the closing of the stoma. So when their stoma closes, they will not do a lot of photosynthesis, and that can be very detrimental for them, especially if there's a long prolonged temperature rises keeps on going. So this could be very detrimental. However, there are other plants that have actually devised many ways to combat this. And these are called C4 and CAM plants. So these plants, they actually use another type of photosynthesis that we call C4, which we'll speak about them later. But both, what they do is that they actually take carbon dioxide and they actually add it to PEP, this phosphoenol pyruvate. I'm quite sure you guys remember PEP. PEP is actually also coming from glycolysis. So they add carbon dioxide to the PEP and they form a four carbon molecule, hence the C4 coming up. Then PEP can be then transported and actually or can be kept there. Because then what happens is that when we need carbon dioxide, PEP can be actually decarboxylated to release CO2. So this is very important because, for example, let's imagine you have a plant and there's a lot of carbon dioxide. So some of it you're going to use for the C3 plants, so for the C3 photosynthesis to create glucose, and some of the carbon dioxide that you don't need right now, you can actually put it in PEP to form this four carbon molecule, just in case. Because what happens that let's imagine temperature now rises, stomas are closed, we actually don't have too much carbon dioxide coming in. So these now four carbon molecules can then release the carbon dioxide they have to keep on fueling this photosynthesis. And this is basically the C4 photosynthesis. So this really helps actually a lot in two actually very two important actually solutions. One is the spatial solution, and the other one is the temporal solution, which we will speak about. So we're going to talk about more the C4 pathway. So here so basically this is the C3. So this is the carbon cycle where carbon dioxide comes into your mesophyll cell, incorporates into RUBP, and then basically, as we know, it goes to the carbon cycle to give us G3P, which then is used for glucose. So in the C4 pathway, it's a bit different. So what happens is that the carbon dioxide is then put into the C4. Then the C4, sorry, CO2 is put into PEP, which gives you a 4 carbon, so C4. Then this C4 can then release CO2 straight into the vesicle to create, again, this carbon cycle. So those are the main differences. So basically, here is that the C3 CO2 is going directly into the vesicle and helping RUBP turning into 3PG. While here, no. First, carbon dioxide is then put into C4. Then C4 is decarboxylated, so carbon dioxide is removed, and that goes then back into the carbon cycle. So that's the C4 pathway. So C4 plants, they use this pathway very well, and it's very important for them because these plants cannot do the carbon cycle in the mesophyll cell. They can do it in the bundle sheath cell. So what happens is that the carbon dioxide here cannot actually penetrate into the bundle sheath cell because it's impermeable. So what happens is that CO2 is given to CO4. So CO2 is given to PEP, which is then given CO4, and then this CO4 can transport into the bundle sheath. So the first thing, it actually helps with the spatial solutions. So let me show you how. So this is your mesophyll cell, and this is your bundle sheath. Carbon dioxide cannot go into your bundle sheath. It just cannot. It's impermeable. So first of all, what happens is that PEP comes up, and then CO2 is incorporated, and then to give you oxalo acetate. This is the four carbon that we were talking about. This is then converted into malate, and this malate is then able to actually transport into the bundle sheath. So it's able to go straight into your Rubisco to this enzyme and give it straight away the carbon dioxide to fuel the carbon cycle. So it helps actually the transportation of carbon dioxide from one cell to the other, especially when carbon dioxide is not able to do so. So this is very important. And this is actually really, of course, it really helps a lot the protection of carbon dioxide in where imagine you again, like I'm saying, in a time where you have a lot of carbon dioxide, Some of it you use to fuel production of glucose. Some of it you actually keep it is this oxaloacetate, and this really helps you always have a certain amount of carbon dioxide, especially when the stomas are closed, when the temperature rises. So this is actually how C4 plants really can basically use this to accommodate to these issues. So the C4 plants, evolved the C4 pathway to minimize photorespiration. So in order to actually minimize the amount of oxygen going to ribisco, they've actually managed to get the C4 to keep the carbon dioxide in a high amounts, especially when the stoma is closed. No worries. We have basically this oxaloacetate, which is basically carbon dioxide with added to PEP. So that is basically we have a big amount of that. So we can always decarboxylate it to generate more CO2. So in order to actually reduce photorespiration, the issue with C4 pathway is that, as you can see here, that when we get basically when of course when malate comes in and decarboxylates, it's going to turn into pyruvate. Then to get pyruvate back into phosphoenolpyruvate, we need to use ATP. So remember here we need basically six CO2s. So imagine this actually is happening. You basically will need actually 12 additional ATPs. You need a lot of actually 12 additional ATPs compared to the carbon cycle. So that's a big issue here is that the C4 pathway uses much more energy. So rather than just using basically the ATP needed in the carbon cycle, you also need ATPs for the C4, which is 12 additional ones. So now let's talk about CAM plants. CAM plants, they do the same thing, actually, this whole C4 pathway, but they're a bit different. So these guys, of course, they fix CO2 into the PEP during the night, and then they store it in the vacuole. So the only difference between here is that they don't have this problem of the spatial. They don't. They do this whole reaction here. They do it in one cell, so they don't have an issue of basically transporting it. Their only issue here right now is the timing, because these plants during the day they close their stomas because it's super hot. They only open the stomas when it's actually quite dark during the night because it's actually less hot. So it actually helps them, still do basically do this photosynthesis or this kind of let's say carbon cycle during the day. So let me use it here. So during the night, they're basically taking up all this carbon dioxide and they're basically, putting it into the PEP to give you this oxaloacetate, the C4. And during the day, when the stoma is closed and there's very little CO2, no worries because basically we have so much of this oxaloacetate that's basically being decarboxylated and giving CO2. So that's the nice thing of the CAM plants. So again, I'm going to repeat this in the C4 pathway. The most important for in C4 plants basically this oxaloacetate really helps it in the spatial problems that the mesophyll cannot do the carbon cycle. It needs to be transported into the bundle sheath cell because carbon dioxide cannot go inside the bundle sheath cell. It's impermeable. So how to do this is when you do this basically the C4 pathway, then you get basically you can transport it. In CAM plants, you don't have this problem because this is all done in the same cell as you can see here, but it helps actually more in the timing because of course these plants they close their stomas really during the day, so they have very little CO2. So to combat that during the night where actually the stomas are open, it's gonna get so much CO2 and create the C4. And this C4 is gonna then be used later in the day to help fuel actually the carbon cycle despite the stoma being closed. So this is how, this C4 pathway has actually helped a lot CAM plants and C4 plants to actually, combat the issue that you have when it comes to having too much heat and the stomach closing. So again, I'm going to show you this here, basically the comparisons between the C4 and CAM. So C4 and CAM plants, both of them use the C3 and C4 pathways. So C3 is the Calvin cycle, and the C4 pathway is basically the PEP, where we get carbon dioxide and we put it to PEP to give us oxaloacetate. So both of them use it. But the C4, the two pathways occur in different cells. The C4 pathway happens in the mesophyll, and then basically the Calvin cycle, the C3 pathway, happens in the bundle sheath cell. The CAM, the C4 and the C3 pathways happen in the same cell, but It's just the timing. C4 happens at night, while the C3 pathway happens during the day. So that's the difference between them. So I'm gonna stop here because this is the end of chapter 8.